Greetings to everyone. Teacher Janet here. It is the start of a new academic year, and I would like to wish a warm welcome to all our new Form 4 students who will be studying biology for the next 2 years. So biology is an interesting subject where we study a lot about the processes that go on in the human body and also about other organisms and the environment. Today we are going to discuss an interesting topic from Form 4 Chapter 2 of the Biology KSSM syllabus entitled Cell Biology and Organization. The subtopic is 2.1, Cell Structure and Function. So, what are cells? Now, cells are the smallest biological unit of life found in living organisms. They are called the building blocks of life. Just as a house is made of bricks, so an organism is made up of many cells. For humans, we are made up of trillions of cells. Now, the interesting thing about a cell is that it is made up of many cellular components within, such as these here, and they all have different functions, but they work together for the survival of the cell, so that the cell can carry out the basic living processes like respiration, excretion, nutrition, even movement for some cells, such as white blood cells and sperm cells. So the human body has about 200 different types of cells, such as muscle cells, red blood cells, white blood cells, nerve cells, with a wide range of structures and functions, and each cell is well adapted to carry out its function efficiently. Let's find out more about our basic unit, the amazing cell. Now, what are the learning standards or learning outcomes that a student must achieve, after this video? So, for topic 2.1, cell structure and function, the first learning standard is you must be able to prepare microscope slides of animal and plant cells. So this is a practical activity in the laboratory, and the teacher will teach you how to prepare the microscope slides of animal cells like the cheek cell, cell PP, taken from the mouth of students, and also for plant cells, usually the plant cells that are observed under the microscope are the onion cells, the epidermal cells of the onion bulb. Okay, so other than preparing these microscope slides, you must be able to draw and label the structures, inside the cells or the structures of the cells. Now, identify the structures of animal and plant cells based on observations through a light microscope. So light microscope is the normal type of microscope that we use in the laboratory, which uses light to illuminate the slide, so that you can see the structures clearly. So this is not a very powerful microscope compared to the electron microscope, which is able to enlarge the object, produce an enlarged image of the object, which is to view the structures very clearly. So we'll talk about that later. Now, number 3, analyze the components of animal and plant cells as seen on micrographs. So micrographs, what are they? So they are photographs taken by using a microscope, like the electron microscope. So micrograph comes from the word microscope and photograph. So a photograph taken by using a microscope, such as the electron microscope, and it is actually a magnified photograph of an object. You can see these pictures in your Form 4 textbook, the micrographs. So you must be able to recognize the components on the micrographs, right? Now, state the main functions of components of animal and plant cells as seen on micrographs, right? This will be discussed in video number 2. We'll discuss the functions of the components of animal and plant cells. So the parts of the animal and plant cells, such as the nucleus, ribosome, mitochondria, and so forth. We'll discuss the structure and the functions of these components, which are mostly organelles. So we'll talk about that later. And then lastly, we're going to compare and contrast the components of animal and plant cells to see the difference between animal and plant cells. So this will also be in video number 2, part 2. Okay, it will be in the second video after this. So first, let's discuss cell structure and function, subtopic 2.1, right? Now, all living things are made up of cells that are combined together. Okay, so cells are like the building blocks of the bodies of organisms. Now, the definition for cells are the basic structural units of all living things, right? That means they are the units that are used to build up the structure of the body of living things or organisms, such as human beings, animals like cats, fishes, insects, even worms, and so forth. They are all made up of cells. Okay, so for a human being, the number of cells in the body is about 30 trillion cells. Okay, right. Next, we have the word organelles. Organelles actually mean, within inverted commas, tiny organs, but they are not organs. Okay, because organs are bigger. Organs are made of many cells. So organelles are the tiny, small structures in a cell which perform specific functions, such as the nucleus, mitochondrion, chloroplast, vacuole, and so forth, which we will study later. Now here you can see a nucleus, right, which has a spherical or round shape. Okay, that's the control center of the cell. And then in plant cells, there is the chloroplast, which is an organelle that carries out photosynthesis to produce organic food substances for the plant. And then cells must have mitochondrion, okay, which is an organelle that is involved in respiration to produce energy, okay, for the cell to carry out its activities. Now, what about the word cellular components? So cellular components are parts of a cell that have different functions. So they will include the organelles. They include the organelles such as the nucleus, mitochondrion, ribosome, and lysosome, and other structures such as cell wall, plasma membrane, and cytoplasm. So these other structures are not called organelles. So the cell wall in plant cells, plasma membrane, and cytoplasm. Okay, they are not called organelles. Now, so cellular components is just a general name for all the parts of the cell. They have different functions and they include organelles. Now, so the cellular components that can be seen under the light microscope, which we have in the school laboratory, right, are plasma membrane, cytoplasm, nucleus, cell wall, large central vacuole in plant cells, and chloroplast. Okay, so take note that the first three, plasma membrane, cytoplasm, nucleus, are found in both plant cells and animal cells. Okay, but the last three here, cell wall, large central vacuole, and chloroplast, are only found in plant cells, not in animal cells. Okay, now we'll discuss more of this in the next slide. Right, so in your textbook, there are two practical activities that you can carry out, activity 2.1 and 2.2, to prepare and then examine animal and plant cells under the light microscope. So this is an example of a light microscope. It has an eyepiece and then a few objective lenses of different power. And then here's the stage for you to put the slide. Then below it is the light source. It could be a light bulb connected to an electrical supply, or it could be a mirror that reflects the sunlight onto the stage. Okay. Then, so the aim of the light microscope is to produce a magnified image of the object that we want to see, and using light to illuminate the stage. okay, to brighten up so that we can see the image clearly. Now, so how do we prepare the cheek cells and what is the structure of the cheek cells? Now, firstly, we have to gently scrape the inside of the cheek with a blunt toothpick, cell PP, get the cell PP or cheek cells. And then transfer the cells on the tip of the toothpick into a drop of water on a clean glass slide. Now, actually, the tip of the toothpick you cannot see the cells on there, so it looks like there's nothing. But actually, once you have scraped the cheek with the toothpick, the cells are already there. So place the cells in a drop of water on a clean glass slide and cover with the cover slip, and then stain the cheek cells with methylene blue solution using a technique called the irrigation technique, which your teacher will teach you. And then examine the cheek cells under the light microscope, and this is the result of our observation. Okay, we have to know how to draw the structure of cheek cells, which is a type of animal cells. So firstly, we can see that cheek cells do not have a fixed shape or a regular shape. So this cheek cell has a different shape from this one. And then there are three parts that we can see: the plasma membrane, which is the outer boundary, then the cytoplasm, which is stained light blue, and the nucleus, which is stained dark blue by the methylene blue solution. In the cytoplasm, you can see some dark spots. Those could be the organelles, or tiny structures within the cell that carry out different functions. So we'll discuss more of the organelles later. So that's all you can see of the cheek cells under the light microscope, which is not a very powerful microscope. If you use another type of microscope called the electron microscope, you'll be able to see the organelles inside the cytoplasm. Okay, more about that later. Now let's go on to the second type of cell, which is the plant cell. So for this, we are going to use the epidermal cells of the onion to observe how a plant cell looks like. Now, onion bulb, okay, you can cut it into half, and then you can see a few layers of the scale leaves. These are called scale leaves, and they are not green in color because scale leaves are for storing organic food substances, okay, produced by the green leaves above the ground. So the scale leaves are all under the ground. The bulb is under the ground, so it doesn't carry out photosynthesis, so there's no chloroplast, no green chloroplast, which is a type of organelle that is green in color and carries out photosynthesis, okay, to produce the organic food substances for the plant. So epidermal cells of onion, remember, of the onion bulb do not have chloroplast, okay, because they're not green in color. So preparation is like this. So first we get a scale leaf from the onion, and then we remove the translucent epidermis from the inside of the scale leaf. So you take out one scale leaf, is the inner side, okay, the epidermis on the inner side, which is translucent, that we take out, not the one on the purple side, not the epidermal cells on the purple side. So add a drop of distilled water on the slide, and then lay the onion epidermis on the water, and then cover it with a cover slip, and then again using the irrigation technique, stain the cells, this time with iodine solution. Okay, now what do we observe under the light microscope? So we can see that plant cells have a cell wall. A plant cell has a cell wall, and underneath it is the plasma membrane. Then you have the nucleus stained dark brown, and the cytoplasm is stained light brown or yellowish brown by the iodine solution. Okay, then you may be able to see the vacuole, which is a structure that contains water and other dissolved substances in the center of the cell. Okay, so that's all that you can observe under the light microscope. Cell wall, plasma membrane, nucleus, cytoplasm, and vacuole. Now, how would the chloroplast look like under the light microscope? So now we are going to discuss the structure of a leaf cell, which is also a plant cell. Now let's discuss how the typical leaf cells would look like under the light microscope. So for the leaf cells, we can use the leaf of Hydrilla species or Elodea species, which are both aquatic plants that live submerged in the water, in the pond water. So why do we use the leaf of these plants? Because these leaves are very thin. They have only a few layers of cells. So we can take out one leaf, put it on a drop of water, on a slide, and then cover it with a cover slip. And that's all. You can see the leaf cells quite clearly under the microscope. Okay, so let's look at the leaf cells of Hydrilla species and Elodea species under the light microscope. Okay, you can see that each cell has a cell wall, and then underneath is the plasma membrane. Then again you have the cytoplasm, nucleus, and vacuole. And the additional structure is the chloroplast, compared to the epidermal cell of onion bulb. Okay, now, so this is the structure called the chloroplast, which you can see is green in color. And it is because the chloroplast, which is an organelle that has a specific function in the cell, it contains the pigment chlorophyll. So chloroplast is the small structure in the cell, so it's an organelle, whereas chlorophyll is the green pigment opovorna, the green coloring substance that is used to absorb the sunlight, so that the chloroplast can carry out photosynthesis. So anyway, remember, this structure is called the chloroplast, but the pigment inside is called chlorophyll. Okay, so we can see clearly because the chloroplast is colored green, and is comparatively big, okay, compared to some organelles. All right, so these are the structures that you can see, or the cellular components that you can see, okay, for a leaf cell that is observed under the light microscope. So we have already discussed the structures of animal cells and plant cells as seen under the light microscope, right? So the light microscope usually has a magnification of 1000 times, which is quite low, all right? And this allows us to observe only the larger cellular components like plasma membrane, nucleus, vacuole, chloroplast, right? However, there's another type of microscope called the electron microscope, which is able to magnify a cell more times, many more times, such as 2 million times, to enable us to see the smaller cellular components, especially the organelles, like mitochondrion, ribosome, lysosome, Golgi apparatus, rough endoplasmic reticulum, smooth endoplasmic reticulum, and centrioles, right? So this type of microscope with high magnification and resolution uses electron beams in place of light to produce the enlarged images of tiny objects. As you can see here, the images will be projected onto the computer screen, and the researcher or scientist will sit here and look at what is projected. So the size of the electron microscope is definitely much bigger than that of a light microscope, and so its price is also definitely higher. And you can find these electron microscopes in universities, right? Not in the school. So we are going to study the components of animal and plant cells as seen under the electron microscope, all right? And we are going to study the structure and functions of these components, especially the organelles. Most of them are organelles, right? So let us first review what the components of animal and plant cells are like, okay, based on what is seen under the electron microscope. I wonder, what are the components of an animal cell? Let's find out. Plasma membrane, cytoplasm, nucleus, nucleolus, smooth endoplasmic reticulum, mitochondrion, lysosome, centriole, Golgi apparatus, and lastly, tiny ribosomes. Plant cells. What are the components of a plant cell? Cell wall, plasma membrane, nucleus, nucleolus, cytoplasm, vacuole, chloroplast, rough endoplasmic reticulum, ribosomes, Golgi apparatus, smooth endoplasmic reticulum, and lastly, mitochondria. So in the previous two slides, we managed to view the 3D model of the structure of an animal cell and a plant cell, right? Now, normally in the exam, you are asked to draw any of the cellular components in a cell. Usually, they will ask you to draw the 2D structure. That means two-dimensional shapes or structures which have the breadth and the length and no depth. So don't worry about drawing 3D structures, but rather focus on being able to draw the structures like what is seen here in this two-dimensional diagram. All right. So here is the picture of an animal cell and the structure within. All right. So we are just going to have a quick look at the components of the cell and then discuss very briefly the functions. Right. For example, nucleus. Nucleus is a big organelle in the cell, and it consists of the nuclear membrane, which is a membrane that surrounds the nucleoplasm inside. The nuclear membrane has pores called the nuclear pore, so that substances can move in and out. And then the nucleoplasm is the medium inside the nucleus. And there's a dark black spot called the nucleolus, okay, with specific function. So what is the general function of a nucleus? A nucleus controls all the cellular activities in the cell. It's the big boss in the cell, and it has chromosomes, which are thin thread-like structures like this, that contain a molecule called the DNA molecule, or deoxyribonucleic acid molecule, which determines the characteristics of the cell, and also is involved in the control of the metabolic functions, right? Now next, we have the plasma membrane that surrounds the cell. So the plasma membrane controls the movement of substances into the cell and out of the cell. Then there's the cytoplasm, which is a gel-like medium for the biochemical reactions in the cells to be carried out, right? Next, we have the mitochondrion, which is known as the powerhouse of the cell, powerhouse, because it generates energy through the oxidation of glucose during cellular respiration, generates energy for cellular activities. Okay, and next we have the lysosomes, which are bags containing enzymes, all right, hydrolytic enzymes that can hydrolyze or break down complex organic molecules like protein and lipids. So these lysosomes can digest complex organic molecules and also break down bacteria and also damage components in the cell, like damage mitochondria. Okay, Then next we have the rough endoplasmic reticulum, which is continuous with the nuclear membrane. And the rough endoplasmic reticulum has ribosomes on it, okay, small particles called ribosomes. So the function of the rough endoplasmic reticulum is that it transports proteins synthesized by these ribosomes, the orange particles here, all right? So it's for transport of proteins synthesized by ribosomes, whereas the ribosomes a ribosome will synthesize proteins, all right? So ribosomes are the site for synthesis of proteins, okay, which are then transported by the rough endoplasmic reticulum, right? And then next we have the smooth endoplasmic reticulum, which is also for transport. So the smooth endoplasmic reticulum does not have any ribosomes on it. Its function is to synthesize and transport glycerol and lipids. It also carries out detoxification of drugs, right, in the body. Now, next we have the Golgi apparatus here, all right, which has a C-shaped structures. So the Golgi apparatus modifies, packs, and transports proteins and carbohydrates, which we'll discuss in the next video. Then we have the centrioles. So centrioles are involved in cell division. Okay, they form fibers called spindle fibers during cell division, but they're only found in animal cells. Centrioles are only found in animal cells and not in plant cells. Okay, and then we may have small vacuoles in the cell, which are bags, like sacs or bags, SAC, sacs or bags, containing certain substances inside. Right? So this is the structure of an animal cell. And we will discuss in more detail the structure and functions in the coming video. Lastly, the structure of a plant cell, right? So as I said, you have to know how to draw the mitochondrion, Golgi apparatus, and so forth. So you can, I suggest that you draw the structure of an animal cell and plant cell as is seen here, because this diagram is well drawn for you, okay, with the accurate drawing of the structures, the various structures, like rough endoplasmic reticulum, smooth endoplasmic reticulum, and so forth. Okay, so you can draw the structure of the plant and animal cell, the last two slides here, in your notebook. Now, going on to the plant cell here, what is the difference between the plant cell and animal cell? Okay, plant cells have a few structures here that are not found in the animal cell. Okay, but plant cells have some common structures like mitochondrion, Golgi apparatus, rough endoplasmic reticulum, nucleus, smooth endoplasmic reticulum, which are similar. All these are same, are found also in the animal cell. Okay. Now, what does a plant cell have that is not found in the animal cell? Plant cells have, of course, the chloroplast. Okay, so the chloroplast is an organelle that's involved in the process of photosynthesis. So it contains the pigments called chlorophyll to absorb sunlight and then convert the light energy into chemical energy, which is stored in the food substances like glucose, right? So I put a star here to say that it is only found in the plant cell, because animals cannot produce their own food. They don't produce their own organic food substances through photosynthesis. So they don't have chloroplasts. Okay. Now, another structure that's only found in plant cells is the big central vacuole in the cell. So this big vacuole is found in the matured plant cells, and it consists of two parts. The membrane surrounding the cell sap inside is called tonoplast, and inside there is the cell sap of fluid, okay, that contains water and mineral salts and so forth, and also sugars, amino acids. So what's the function of this big vacuole in the plant cell? It stores the water, sugar, mineral salts, and mineral salts in the cell sap. And also, when it is full of water, the vacuole will press on the cell wall of the plant cell, and make the plant cell become turgid or firm and hard. So this will give support to the plant cell, and also overall it gives support to the plant, right? We'll find out more about the function of vacuole in the next lesson. And lastly, plant cells have something else called the cell wall, okay, the outer layer called the cell wall, which is not found in animal cells. So the cell wall is made of cellulose, okay, which is a tough substance, and cellulose is a type of polysaccharide, right, in the same group as starch and glycogen. The polysaccharide made up of simple units of called the glucose, or glucose molecules. So what is the function of the cell wall? So it is rigid and tough, so it helps to maintain the shape of plant cells. So plant cells have a fixed shape, for example, a rectangular shape, and the cell wall also provides support to the plant cells, right? Okay, so other than that, the other organelles, the function of the other organelles have been mentioned. So plant cells have a big vacuole, chloroplast, and cell wall that is not found in animal cells. The rest of the organelles that we've discussed are found both in plant cells and animal cells, okay, such as mitochondrion, Golgi apparatus, rough endoplasmic reticulum, nucleus, smooth endoplasmic reticulum. So that's all for this lesson. Do share, like, and subscribe if you find this lesson useful. And we will meet again in the next video when I discuss the functions and the structure of the organelles in more detail. So this topic is important for exam. Usually comes out in the structured questions for SPM. Goodbye for now and take care.